Good afternoon, everyone, and thank you for being here. We are going to speak to you today about the International Mouse Phenotyping Consortium and about its large-scale effort to uncover gene function. Speaking today is myself, Violeta Muñoz Fuentes, and my colleague, Hamed Hasseli. We are part of the mouse informatics team at EBI, a team of 10 people, including biologists, software engineers, and bioinformaticians. The IMPC website is mousephenotype.org, and please feel free to look along. So the IMPC is an international consortium, the goal of which is to make knockout mice of virtually every protein coding gene, and that is about 18,000 genes. We focus on poorly characterized genes, or genes about which very little is known, the so-called ignorome. And currently we have almost 7,000 knockout lines produced and characterized. In the map above, you can see the distribution of the centers. You can see that they are worldwide distributed. And these are the world's leading mouse research institutes. The organigram on the right shows our organization and structure. So the mouse clinics, which are distributed worldwide, produce and phenotype the mice. This data is informatically integrated by the Data Coordination Center, which also performs the data validation and quality checks to make sure that all the data makes sense, and they perform the preliminary statistical analysis. The core data archive does additional quality checks, does the final statistical analysis, and deploys all this data in our website. And in this way, we make it available to people like you and to the scientific community or any interested parties. We have, in addition, two resources. One is IMITS, that is used to keep track of the mice that are produced and phenotyped. And the other one is IMPRESS, which collects all the procedures and protocols we apply to the mice to phenotype them and all the parameters that are collected. So we'll now move on to see how the data is generated. First, the knockout mice need to be produced and validated, and then they are phenotyped. And this includes not only the knockout mice, but also the wild-type mice that are used as controls. We apply standardized phenotyping pipeline that are the same across all centers at the embryonic, early adult, and late adult stages. We analyze the data using our custom statistical pipelines that are developed to accommodate high throughput phenotyping data, in which knockout mice are compared with wild-type mice. And then if any phenotypes are significant, these are annotating using the mammalian phenotype ontology. And in the next minutes, we're going to see all of this in a little bit more detail. So as I mentioned, the mice are phenotyped at the adult and embryonic stages. And the pipelines are standardized across all IMPC partner centers. The early adult pipeline that you can see here is applied to all mice, mutants and controls. And depending on the outcome, selected lines are phenotyped in a specialized embryo pipeline. Or some of the lines are also selected for aging. And then a late adult or aging pipeline is applied. So looking at the early adult pipeline in a little bit more detail, it starts with performing a viability procedure, and then seven males and 7 female mutant mice enter the early adult pipeline together with wild types. This is a comprehensive pipeline that covers multiple physiological systems. And then the deviant parameters that are detected inform us about the physiological system that is disrupted when a gene is disabled. And in this way, we can link a gene with function. So these procedures can be grouped together to inform about a physiological system. So all procedures are performed between week 9 and 16, measured in age of a mouse. And for example, the echo and ECG procedures in week 12 can be grouped with a heart rate procedure performed in week 16. And in this way, we learn about the cardiovascular system, and so on for other procedures. So we can learn about neurological or metabolism, and this is represented by these icons that we use in our gene page. So, for example, this is an example for of a gene page. In this case, is CIB2. We have a phenotype summary with icons here on the right top corner of the page, and in orange are the significant significant hits when we find a significant phenotype in a knockout line, and not significant in blue. So this is just a quick look at what the results will tell us in more detail below. So the next thing we can see is a table showing the significant phenotypes, and these are annotated based on the mammalian phenotype ontology, which is a controlled vocabulary of terms, so that no information is lost, and also it can be treated informatically. And I would like to point out that there is a function here, a little app to log in to follow a gene. And in this way, you can register interest in a particular gene, and this information is taken into account by the production centers. By clicking on any of these results, we can have a look at the data in more detail, and thus we give information about all the individuals that were phenotyped, the testing protocols and the measured values and the associated phenotype. You can see all the raw data. It can be downloaded, and you also see the results, the statistical results. So the early adult phenotyping pipeline, as I mentioned, starts with a viability procedure. And this procedure determines whether the homozygous knockouts are viable or are lethal. And if the homozygotes are viable, these are the mutants that enter or that are phenotyped at the early adult pipeline. If the homozygotes are lethal, they are further studied in a specialized embryonic pipeline. And in that case, the heterozygotes of the embryonic lethals will be the mutants that are phenotyped in the adult pipeline. So the embryonic pipeline is applied to approximately a third of the lines, which are the ones that are embryonic lethal. And this is a specialized pipeline that has been created to understand the structural changes that take place during embryonic development. It's mostly a collection of images, and also there's also histopathology and grass morphology procedures applied. This is showing the type of data we are collecting in the embryo pipeline. We determine the effects that occur during development by assessing 2 and 3D images. You can look and compare the mutants. Sorry, I have this. Okay. Yes, you can look and compare the mutants just side by side at the same time. And for example, here we have a 2D image of a wild type and the mutant. You can see the effects in the spinal cord indicated by this arrow. You can see an enlarged thymus indicated by these asterisks, and you can see the effects in the heart. We have the 2D images. This is an interactive viewer, so you can move them around and look at them from different angles. We also have throughout the website, in the specific gene pages, LAC expression. So we can see where, so we can see the tissue in which the gene is expressed. In this case, this is RSPO1 heterozygotes, and we can see expression in the epididymis. So we not only say that a mouse is embryonic lethal, but we can now link gene function to organ systems. And we are also developing automated tools to detect volumetric changes, which is very exciting because this means that we don't need to manually annotate the images, which is quite a slow procedure. So in the next slide, we can see an example of a 3D image view. Again, this is also interactive. So if you go on to the website, you can rotate the images and look at them from different angles, which is quite exciting. So as I mentioned, there's also the late adult phenotyping pipeline. This pipeline tries to understand the effects of aging. The mice are aged until they are 58 to 80 weeks. The adult pipeline is then repeated and the specialized statistical analysis is implemented. And we can see these phenotypes also in the gene page. So there is the old data table, which has specific filters. And you can, for example, filter for late adult phenotypes, or embryo or early adult or mid-adult phenotypes, or you can select them all. And we also have a specialized heat map in which we show all the genes that have been studied in the late adult phenotyping pipeline so far. In orange, you can see significant differences, the procedures that show significant differences between the late adult controls and the late adult knockouts, and in blue, the ones with no significant differences. And by clicking on each of these rectangles, you can look at the underlying data. So I'm now going to pass on to Hamed, who will tell you more about the statistical analysis that we are conducting at the IMPC.
Thank you very much, Violetta. In the next few slides, I will be describing you the stats pipeline in the IMPC. Basically, IMPC Statistical Pipeline is a tool to detect any phenotype effect in the animals that we phenotype. I may use the word phenotype or genotype interchangeably, as they are basically the same thing in the stats pipeline, because we have all the animals that we have are clone of each other, and any difference in phenotype is due to the difference in genotype. So let's go to the stats pipeline. The stats pipeline starts by measuring some knockout animals, normally 7 males and 7 females. And they are normally compared with more than 7, sometimes thousands of controls or wild type or baselines. These data are fed into the stats pipeline, and stats pipeline determines if there is any significant signal in the difference of the knockouts and wild types spotted or not. If we find any significant signal in the difference of between the animals, the stats results are passed into the second pipeline, which is the annotation pipeline. In the annotation pipeline, we select a proper mammalian phenotype ontology term and assign it to that phenotype. So as the result, the stats pipeline output is an MP term, p-value, and a conceptual object that we call in the stat packet. Well, the stat packet describing that is beyond this webinar, but generally is the mean to facilitate communicating between the data scientists, web developers, and other part of the consortium. So in the next slide, we go, we go a little bit deeper into the stats pipeline. IMPC stats pipeline is different from the other pipelines in some aspects. The first and the most important one is the scale of the data that we have. As you see on the left-hand side chart, we the chart on the x-axis, it shows the IMPC data releases from the first release in 2012 to the latest release in 2020, and the y-axis shows the number of data points in million. Here you see three main categories of the data. We have categorical data, like the normal, abnormal, yes, no, and these kind of responses, and time series and non-time series data. We have more than these. We have images, we have human curated data, but in the stats pipeline, we only analyze the three categories, and like images have their own image processing pipeline. As you see in this chart, the data release 11 in 2020, we have an overall of the blue dashed line is the sum of three categories. So we have more than 45 million data points to process by the stats pipeline. So this shows the scale of the IMPC project, that we work with a lot of data. And I should point out that in the next release, we have something, including the images, we have something like 18 million data points, which is a lot. So a stats pipeline should be scalable in IMPC. As you see, the scale of the data is quite increases quite fast. And of course, any stats results should be reproducible. These are the priorities of the stats pipeline by IMPC. However, we have the accuracy is important for us. Accuracy here points to the applying the most accurate model or test, statistical model or statistical test to the data. And the whole stats pipeline should be reliable in the aspect that it shouldn't fail. It should cope with different and diverse scenarios in the data without failing, as any failure in the pipeline is very difficult to trace back and fix. So for us, coming back to the accuracy, we also try our best to apply the best accurate model that we could to the data. For this sake, we have designed and developed a new methodology. We call it soft windowing, that I will explain in the next slide. Yeah. Soft windowing is a methodology to reduce the false positives in the stats results, and of course, it's one of the other features of that is to make consistent results between the IMPC data releases. But what's the soft windowing and how it works? Soft windowing is a method to window the data, but window the data with respect to the mutants, with respect to the knockouts. Here in the scatter plot, you see the blue dots versus the orange dot. The blue dots are the mutants, and the orange dots are wild types. As I pointed out in the first slide, we normally have 7 mutants, males, and 7 females, and we could have thousands of controls, as you see in this chart. And you see that the time span here is from January 2013 to January 2017, and you see the fluctuation of the data over time. Basically, we don't know what's the cause of these fluctuations. It could be the change in experimenter, it changed in the many factors, like change the lighting of the labs, or even changing the perfume of the experimenter could end up a trend in the data. But how soft windowing works is to find the most proper controls around this mutant, as you see is kind of like blocked in the kind of like a little window that you see here. There is a little video that if we could see that, it shows a very basic functionality of the soft windowing. Yeah, soft windowing, here you see two vertical lines. These are the mutants, the imaginary mutants, and the window starts expanding and expanding, and if there is any merge or intersection between them, it merges. In this step, this is the second step of that. Basically, soft windowing starts to find the best match window around the data. I should point out that like window in the wind, it was so-called soft windowing is not a hard threshold. It could be very soft. It's why we call it soft windowing. Okay, going back to the next slide. Yeah, here it shows the overall workflow of the IMPC. Everything starts from a protocol. Protocol is nothing more than the design of the experiment. From that protocol, there are some measurements, which we call them parameters. And for each parameter or each measurement, we have some data. And these data are analyzed statistically, and from a set of predefined ontological terms, we choose the best one based on the stats results. In the next slide, I just like introduce you some of the open source softwares that we use. So the statistical engine of the IMPC statistical pipeline is a software written in R. Is called OpenStats. It's an R package, and it's available freely from Byconductor. For soft windowing, if you are interested, there is another R package which is hosted on CRAN. We have the publication of soft windowing here. OpenStats publication is under review now. If you are keen to run the stats pipeline, IMPC stats pipeline, or use a part of it, or anything that you are interested, the entire stats pipeline, IMPC stats pipeline, is available from GitHub, annotation pipeline as well, and we have a little wiki page for that. If you are a fan of Docker containers, we have the Docker container of the stats IMPC stats pipeline available from the Docker Hub. And I guess I'm done. If you have any question in the end of the webinar, I will be happy to answer that. So I pass on to Violetta for the rest of the presentation. Thank you.
Thank you very much, Hamed, for this introduction to the IMPC statistical pipeline. So we are now going to move to key findings and achievements. And the first study I would like to talk to you about is a study published in Nature Communications, which shows the wide extent of sexual dimorphism, both in wild type and knockout mice. And the data I'm going to present, the pie charts, represent continuous characters, but we also see the same for categorical characters. And in this figure, you can see that for almost 40% of the parameters we've measured, the phenotype depends on the sex. And for 21% of the parameters, the males present greater values. And for 18%, it is the females that present greater values for whichever data it is that we were measuring. So we also see that for the knockout lines, for 12% of the parameters, not only one sex presents the phenotype. And interestingly, for 4% of the parameters, we see that males and females show opposite phenotypes. So in order to, so I will show you an example. So this is a knockout strain for the gene GALD18. which shows opposite sexually dimorphic phenotypes for fructosamine, which was measured in the plasma. And as you can see, here are the female mutants in blue, which present an increase fructosamine concentration as compared to the wild types. And for the male mutants, we see a decrease in fructosamine levels. So the IMPC demonstrates the effect of sex on many phenotypes, and these results have led to an exciting dialogue and to reports in the media, and supports that researchers need to consider both genders in biomedical experiments. The IMPC strives to ultimately support diagnosis of human disease. And we are collaborating with the Monarch Initiative to develop integration of terms in the human phenotype ontology, which used to describe patients, with the mammalian phenotype ontologies that are used to describe abnormalities in mouse strains. So for example, you can find an equivalence between hypoplasia of the maxillae and short maxillae, and the cross-species phenotype would be maxillae hypoplasia. And this is true for morphological characters, but also for physiological characters, such as, for example, decreased circulating cholesterol level. And in this way, we can link human diseases to mouse models. So this is a study that was published in Nature Genetics, in which 3,328 genes that were phenotyped by the IMPC were surveyed. And the semantic phenotype overlap of mouse strains and human diseases was demonstrated, and 360 new disease models were proposed. The list is in this paper, and we are now going to look at an example, which is a gene called PAM53B, about which basically nothing was known before this data came along. So IMPC phenotyping reveals an increase mean of the volume of the red blood cells, and this is depicted in this figure. So you can see for both the female and the male mutants, there is an increase in the volume of blood red cells, and we also see a decrease in the red blood cell number, which is shown in this figure. So again, the phenotype is presented by both female and male mutants, and there is, as you can see, a clear decrease in the number of red blood cells. And this is quite important because the number, volume, and the shape of the red blood cells is critical and needs to be tightly regulated. And this mouse strain presents a good model for anemia. So the IMPC not only provides data, but also models that researchers can further study and then follow them and perform additional experiments. We are striving to understand embryo development, and this study was published in Nature, in which we look at embryo viability data. A wealth of image data was collected, including X-ray, LAC-Z, histopathology, embryo 3D images. We have in the portal an embryonic landing page that you are very welcome to view. It has impressive images, and there's also the interactive embryo viewer I mentioned earlier. With this study, we could associate genes to developmental phenotypes. And we also show that human disease genes are enriched for essential genes. Essential genes are the ones that the homozygote knockouts present a lethal or a viable phenotype. And we further went to study these essential genes in a follow-up study. So in this study, we combined IMPC mouse viability and phenotypic data with human cell essentiality screens. And these human cell essentiality screens, basically what they are doing is knocking one gene at a time, knocking out one gene at a time, and then seeing if the cells survive and proliferate, or if, on the contrary, they die, obviously by the absence of this gene. So, so using this this combination of data, we could classify the genes into 5 mutually exclusive categories, from more to less essential, and we call this fusel binning. And fusel stands for the full spectrum of intolerance to loss of function. In this figure, you can see that the genes classified as viable by the IMPC, which are the ones in blue, and the genes classified as viable by the IMPC, which are the ones in yellow, have a human ortholog that is non-essential in human cell lines. So these bins here, they represent the essentiality in human cells. And in the y-axis, you can see the number of genes. However, for the lethal genes, it's quite interesting because for those genes classified as lethal by the IMPC, we see that a proportion of them have a human ortholog that is essential in human cells, but we also see that a proportion of them have a human ortholog that is non-essential in human cells. And interestingly, these genes here in this part of the spectrum we call developmental lethal, and so that is the genes that are essential in mice but not in human cells. And we see that they are enriched for genes associated with developmental disease, and this is what this figure is showing. So here we see the 5 bins. So these are the viables, so the 5 fusel bins. So these are the viable category, the cellular lethal, subviable categories, and these are the developmental lethal. And you can see that the enriching in developmental disease is quite striking. And therefore, we demonstrate that fusel is a useful tool for disease gene discovery. And in this paper, we identified 163 genes that could be novel candidates for developmental disease. One such gene is VPS4A, which had not been previously reported as having pathogenic variants. However, 3 variants were seen in this gene in 3 patients of 2 rare disease consortiums. So one is the 1000 Genomes Project, and the other one is the Center for Mendelian Genomics. And all these patients presented intellectual disability. And there was also a high overlap of these and other phenotypes between the patients. The orthologous mouse gene, which has the same name, VPS4A, we have a mouse knockout that was phenotyped by the IMPC, and the homozygous were all lethal. The heterozygous had eye phenotypes that were also seen in patients, and the homozygous embryos had brain and other abnormalities. And the GTEx and mouse expression data show that this gene is widely expressed in the organism. We are also translating to other species, and because of obviously genes that are essential in mice are likely indispensable in other species as well, especially mammals. So we have conducted a small study, a pilot study, to show how this could be done in practice. The idea behind this study is that deleterious mutations could be identified in individuals that may compromise fitness or could be associated to disease. And in this way, deleterious mutations could be avoided by selecting the best founders in, for example, in breeding programs for endangered species. The IMPC can also suggest candidate genes by selecting genes that are associated with relevant phenotypes, for example, fertility, metabolism, or pigmentation, and then looking at the orthologs in these species. Or the data also can, IMPC data can also be used to support function, for example, when studying adaptive features. So as an example, this figure shows, so on the x-axis, you can see the number of genes with significant phenotypes, and on the y-axis, you can see a series of phenotypes associated with reproduction. And this figure basically showing that there is a high number of orthologous genes between other species and the mouse. So the mouse represented here in black, and the other species, in this case, we did this for giant panda, gray wolf, western gorilla, Iberian lynx, Tasmanian devil, and polar bears. So showing that there is a high number of orthologous genes between other species and the mouse, and in this case, for genes that are associated to reproduction phenotypes. We are also quite excited about this study, in which we are applying machine learning to automatically annotate X-ray images. Deep learning was applied to X-ray images of wild-type females and males, and this can now be applied to automatically annotate gender in knockout strains, so that abnormalities can be detected in sex-related phenotypes. Again, this automated annotation of images is very exciting because the manual annotation is cumbersome and takes a lot of time. There are other IMPC key achievements that I won't have time to talk about in detail. We are detecting extensive pleiotropy. We are conducting systematic analysis published on metabolism, hearing, eye. There's more underway. A hearing study revealed 52 genes associated with deafness for the first time. And this is important because the vast majority of genes associated with hearing loss are unknown. And in a metabolism study, 21 genes that had not been previously linked that were annotated, that were not previously linked to metabolic disorders. We have also now more than 1,500 CRISPR modified lines, and this is providing insight into mammalian genotype editing, genome editing. This is a gene that was associated to a metabolic disorder, is RSVN1. As you can see here in blue, you see the body weight for males and females. And in orange, you see the body weight for wild type males and females. And also depicted in these figures is the remarkably increasing weight of the male heterozygotes as compared to the male wild type. And we also see an increase in the levels of triglycerides. So the mutants, we see this for both female and male mutants, and in orange you see the levels for the wild types. So in this case, the homozygous mutant was lethal, and that is why it was a heterozygote that was analyzed in the early adult phenotyping pipeline. So we now move on to talk a little bit about impact and integration. And there is, so this is a very busy slide. I won't go into details, but this is to show that there is in-depth secondary phenotyping conducted of IMPC mice. 4 papers appeared last year, and in 2 of these, more than 1,000 IMPC genes were characterized. And they were in-depth characterized for neuroanatomical parameters, for auditory brainstem response, for immunology phenotypes, and in-depth bone phenotyping. We also collaborate with many other consortia And one very important activity is the support of the diagnosis of rare disease patients. Here you can see some of our collaborating partners and institutes. This is not an exhaustive list, and I welcome you to further explore this in our website. The IMPC mice can be ordered. They are archived after phenotype is completed. And the ordering information can be found through the order section in the gene pages. And there are two main repositories for IMPC mice. One is in the US, UC Davis, the UC Davis Comp repository, and the other one is in Europe, the Infrafrontier repository. The consortium tracks publications that use our mice or our data, and this is done by our team, but also we feed from mouse genome informatics curated publications. We see an astounding number of currently 3,264 papers, which basically is indicating that people are using our strains and our data. And obviously, this is, of course, very exciting for us. So just to finish, concluding remarks, the high throughput IMPC phenotyping is revealing thousands of new gene phenotype associations. We are connecting clinical phenotype data with mouse phenotypes, and we are also coordinating mouse production across rare disease consortia to support the diagnosis of these patients. We are developing a new tracking system that we hope it will be released very soon, maybe in the next weeks or in the coming month, to accommodate all types of plant-induced mutations. It will be called GeneTAR, and it will replace IMIDs. We are continuously looking for the best ways to provide feedback and communicate the design, the production, and our phenotyping results. We are happy to get involved in new collaborations, and all data is freely available at mousephenotype.org. The project is ongoing, so please keep looking. And I just need to finish by acknowledging a great number of people. It's just two of us here today representing a tremendous group of people. And I don't have a listing of all the funding, but some of the main ones. And the informatics team is funded by the NIH. I thank you very much, and we hope you enjoy looking at our resources.